Hello dear candidates, welcome again to Bioclasses online. As you know, we have started the post-Mendelian inheritance and we have already discussed the two types of the gene interactions, that is interallelic gene interaction and non-allelic gene interaction. We have also discussed one of the example of the interallelic gene interaction, that is incomplete dominance. The link of that video is in the description. And today we are going to discuss about the second example of the interallelic gene interaction, that is codominance. Dear candidates, if you have not subscribed my YouTube channel yet, do subscribe now and hit the bell icon so that you will remain updated with my channel, that is Bioclasses online. Now without wasting time, let's start. Codominance is the condition where both the alleles of a gene express themselves simultaneously in a heterozygote. Or in other words, we can say when both the alleles of a gene express themselves simultaneously in a heterozygote, this condition is called as codominance and the alleles are called as the codominant alleles. For example, if there are the two alleles, allele A and allele B, and if these alleles are the codominant alleles, then that means that when these alleles A and B are together in an individual, both these alleles express themselves simultaneously in an heterozygote, and these allele A and allele B are the codominant alleles. These codominant alleles neither show dominant recessive relationship nor they show the intermediate condition, but express their traits independently. The symbols used for codominant alleles is also different. Here the codominant alleles are mainly shown by the same capital letter with the different subscripts. For example, the codominant alleles of AB blood group are denoted by IA and IB. The another method to show these codominant alleles is by their own capital alphabets. For example, the red hair color in cattle is denoted by an allele capital R, while as the white hair color in cattle is denoted by the allele W, capital W. Now let's take few examples of this codominance. The first example of the codominance is the AB blood group. Before we discuss this AB blood group as a as an example of a codominance, let's first share some information about the ABO blood group. As we know that there are 4 blood groups in human beings. The blood groups are A, B, AB and O, and out of these 4 blood groups, the blood groups A, B and O were discovered by the Landsteiner in 1900, while as the blood group AB was discovered by De Castillo and Stanley in 1902. And the one more important point regarding these blood groups, that is the ABO blood group, is that the plasma membrane of RBCs, that is the red blood corpuscles, has glycoproteins. These glycoproteins are called as antigens or agglutinogens or isogglutinogens. So the plasma membranes of the human RBCs has glycoproteins called as antigens or agglutinogens or isogglutinogens. These glycoproteins or these antigens are controlled by a gene denoted by the capital I. And this gene I has the 3 alleles, IO, IA and IB. And out of these 3 alleles, IA and IB are the dominant alleles, while as the IO is the recessive alleles. So IA and IB are the dominant alleles, and if they are present together, that means both these alleles will express themselves simultaneously in a heterozygote condition. That means these are the codominant alleles. And while as the IO is the recessive allele and is not able to express itself in presence of IA or IB. And I antigen, sorry, allele IA will produce an antigen called as antigen A, while as the IB will produce the antigen antigen B. But the allele IO does not produce any surface antigen, that is neither A antigen nor B antigen. And one important point about this ABO blood group is in every person only 2 of the 3 alleles may be present. Out of these 3 alleles, any of the 2 alleles may be present in every person. Now let's try to see in the table that about this ABO blood group. As you can see here that the individual who's having the genotype of IO, that means both the recessive alleles are present in an individual. In that case, neither of the antigen, neither the A antigen nor the B antigen will be present on the plasma membrane of RBCs, and such individuals will have the blood group of O. So this is a case of the recessive condition, that is the IO is the recessive allele, and when they are present in the homozygous condition, that means the blood group is O. The second genotype may be, that is IA or IAIO. If this is the condition, either the IA or IAIO alleles are present in an individual, in that case the blood group of an individual will be A, because the antigen on the surface of the plasma membrane will be A in both the cases, because in IA both the alleles will get expressed themselves, because both are the dominant alleles and will form the antigen A. And in this case, that is the IAIO0, IA is dominant over the IO. So IA will express itself and will produce the antigen A. And this is the case of the dominance or the dominance case, that is IA is dominant over the IO. The third case is, that is if the genotype of an individual is IB or IBIO, same as in case of the IA, the antigen will be the B antigen, because B allele IB will produce the surface antigen on the plasma membrane of RBCs. This is again the persons will have, and an individual will have the blood group of B. And this is also the case of the dominance. Whereas if the individual have the genotype of IA and IB, that means both the dominant alleles are present and both are able to express themselves simultaneously, and that means IA will produce the antigen A and IB will produce the antigen B. So in such individuals, both the antigens, that is antigen A as well as the antigen B, will be present on the surface or on the plasma membrane of the RBCs, and the persons will have the blood group of AB. And this is one of the classical example of the codominance, because here you can see that IA as well as the IB alleles are expressing themselves simultaneously and are producing both the antigens, that the antigen A as well as the antigen B on the plasma membrane of RBCs. So you can see here now that only this IA and when the individual is having the blood group of AB, that means the alleles present in that individual are IA and IB, and both these alleles are expressing themselves simultaneously, and both are expressing themselves simultaneously, and that means that both the antigens A and B antigens are present on the plasma membrane of RBCs in such individuals, and this is an example of the codominance. And this ABO blood group is also an excellent example of the multiple alleles. Multiple alleles because the three alleles govern the same character. When more than two alleles govern or responsible for a particular character, such a condition is known as multiple allelism, or multiple alleles are such alleles are called the multiple alleles. Here you can see in case of the ABO blood group there are three alleles, IA, IB and IO, and this is the condition in ABO blood group. IA and IB alleles are the codominant alleles, while as there are three alleles responsible for this character, that is the ABO blood group, or that is a case of the multiple allelism. And so therefore we can conclude here that the alleles for the blood group A and blood group B are codominant, that is the IA and IB are the codominant alleles, so that when they come together in an individual, as I already mentioned, when they come together in an individual, they will produce the blood group of AB. It is characterized by the presence of an antigen A as well as the antigen B over the surface of the RBCs. Now let's try to understand the inheritance pattern of this codominant alleles. If an individual is having the blood group of A, B, and the second individual is also having the blood group of AB, the alleles the gametes they will produce, that means the gametes they will produce the IA and IB. One individual will produce the IB gametes carrying the allele IA and gametes carrying the IB. The same way here the two types of the gametes will be formed. One of the type of the gamete will be having the IA and the second type of the gametes will be having the IB. And if you will cross here, that if this is crossed here, you will get the IA, and if this IA is crossed with this one, you will get the IB. So this will be the blood group A, this will be the blood group AB, and if this IB is crossed with this, you will get IA and IB, and if this is crossed with this one, you will get IB. So you can see here we are getting the IA1 and IB2 and IB1. So the ratio is 1:2:1 here when an heterozygote for the codominant alleles are crossed with one another. So here the ratio we are getting the 1:2:1. Now let's try to understand this codominance with one more example, that is the sickle cell anemia. This, as we know, sickle cell anemia is a hereditary disease found in the tropical Africa, but is now spreading in the American blacks whose ancestors have come from the parts of the Africa. In India, if we will take in India it is found in some parts of the Kerala, and the person suffering from this disease, that is the sickle cell anemia, have sickle cell hemoglobin, and the sickle cell hemoglobin, the allele for the sickle cell hemoglobin is denoted by the HbS, and such RBCs become sickle shaped. The RBCs having the sickle cell hemoglobin, that is the HbS allele, such RBCs will become the sickle shaped under low oxygen condition or low oxygen concentration. And if we will say this disease, sickle cell anemia, is caused by the gene HbS, and its normal allele is denoted by the HbA, which produce a normal hemoglobin. And here we will see here the persons with having the genotype of HbA have a normal hemoglobin, but the persons with HbS have the sickle cell hemoglobin, and these individuals that is having the genotype of HBs will die of the lethal or fatal anemia. But the heterozygous persons having the genotype of HbS genotype have both types of the hemoglobin and suffer from a mild anemia because some of their RBCs during oxygen deficiency become sickle shaped, and it shows that the HbA and HbS genes are codominant. Now let's see the inheritance pattern of this sickle cell anemia. If we have the two individuals which act as the sickle cell carriers, or they are the heterozygous, that is they possess the genotype of HbA, HbS, and you can see here the sickle cell carrier or the heterozygous having the HbA, HbS, HbA, HbS, and they will produce the two types of the gametes. This individual will produce the one of the type of the gametes carrying the this individual will produce the gametes, that is the one of the type of the gamete having the HbA, one of the type of the gamete carrying the HbS allele. In the same way, the second carrier or the second heterozygous individual will produce the same type of the gamete having one of the type of the gamete will be having the HbA and another one will be having the, and if we will cross here we will get normal 1, sickle cell carrier 2, and anemic which dies having the genotype of HbS 1. So again here you can see here that the ratio is 1:2:1. Now let's take the one more example of this codominance, that is the coat color in cattle. In cattle, gene R stands for the red color, while as the gene W, capital W, for the white color. And when these red cattle are crossed with the white cattle, that is when homozygous red cattle are crossed with the white cattle, that is the capital R is crossed with the capital W, the hybrids of F1 generation are of roan color with genotype of capital R W, capital W. The roan color in the cattle comprises dark color, that is red, interspersed with the white color which develops side by side in the heterozygous organism. In F2 generation, red, roan and white cattle are produced in the ratio of 1:2:1. That is 1 having capital R, 2 having the genotype of capital R, capital W, 1 having the genotype of capital W, capital W. So the roan color cattle are heterozygous, which shows here that R gene and W gene are the codominant genes, codominant. And you can see here in this diagram that when the red color cattle is crossed with the white color cattle, as we know that the red color cattle are denoted by the capital R alleles, and the white color cattle are denoted by the capital W. The two types of the gametes carry will be the capital R, capital W. And if they are crossed in an F1 generation, you will get the roan color cattle, neither the red nor the white. So there is not the case of the dominant recessive condition. So when they capital R allele and the capital W allele are present together, they express themselves simultaneously. That is why the roan colored cattle are formed. And when these heterozygous roan colored cattle are allowed to cross with one another, you can see here we are getting in the F1 generation having the red colored cattle having the genotype of capital R 1, and roan colored cattle having the genotype of capital R W 2, and white colored cattle having the genotype of capital W 1. So again here we are getting the 1:2:1 ratio. So when the codominant alleles present in the heterozygous conditions are allowed to cross with one another, we are getting the ratio of 1:2:1 ratio. That is the F2 generation ratio in codominant alleles is 1:2:1. Subscribe and share Bioclasses online. Thank you.